So, tá fáilte roimh go léir an seo an hocht, go dtí an seomra starúil seo. I think the last time there was a meeting in this room, it ended in a civil war. Some of you may not be aware that this is a historic chamber. It was the site of the first Dáil, the first Irish Parliament meeting in 1919. So it's remembered fondly in this country as a place where Irish democracy began to find its first full expression within the country. So you're very welcome here tonight for what has become a very strong part of the Teagasc approach to science, and that is sharing and discussing complex issues, some of them difficult issues for people to discuss and arrive at conclusions, but all of them very important and central to not just our own sustainable existence, but also, very importantly, to the sustainability of life for people throughout the world. This is the first in a series, and we'll be hearing in a moment the rationale behind it. But I want to acknowledge as well the number of students who are here. I know some of you have travelled from Moore Park and perhaps other centres, despite the fact that you had events till quite late this afternoon. So we particularly appreciate the presence of students here this evening. So without really any further ado, I'd like to call on Frank O'Mara, Dr. Frank O'Mara, the Director of Research at Teagasc, to give us a little bit of a background to what we're about to hear. Thank you.
Thank you, Leo, and good evening, everybody. And I'd like to welcome you all, that's members of our authority, colleagues and former colleagues in Teagasc, staff from the Department of Agriculture, Food and the Marine, and all others distinguished in your own right, I'm sure, to this lecture here this evening. And I look forward to an interesting evening in your company. So just by way of background, this is the inaugural lecture in a new series of distinguished lecturers, pardon that pun, that Teagasc is initiating this evening. Some of you will know that we had a lecture series that concluded last July with a lecture here in this room given by the Minister for Agriculture, Simon Coveney, around the grand challenges facing agriculture. And that was a very successful lecture series, we felt. And I think we're all aware of the big challenges facing agriculture, and things like food security and nutrition security and climate change and energy security, sustainability, all those things. So there are many big issues facing agriculture, and our previous lecture series dealt with those big issues. But I suppose, when we got to the end of that, it's obvious that technology is going to have to play a role in how agriculture can respond to those challenges. So this lecture series will aim to present new developments in science and technology that have the potential to improve agriculture and to stimulate debate on these new technologies, certainly in an Irish and in an EU context. So we're kicking off this new series of annual lectures. We hope to bring every year a very distinguished scientist to these shores to speak on a particular topic that's of relevance to technology and agriculture and food production. And we're starting this evening with Professor Ford Denison, who is an adjunct professor in ecology and evolution at the University of Minnesota. And I suppose Ford has come to the attention of the scientific community, I'm sure, for many years, but in particular since the publication of his book, Darwinian Agriculture, How Understanding Evolution Can Improve Agriculture. I think it's a fantastic title, that really starts you wondering, what's this book about? So it certainly is a title that captures your curiosity at least. And Ford, his career in brief, I'm not going to go through every piece of it, but he studied ecology at Evergreen State College, Washington, and then earned a PhD in crop science from Cornell. And over his career, he spent time with the USDA, United States Department of Agriculture, and in University of California, Davis. And he has ended up at this stage of his career in the University of Minnesota. And most of his research focused on the symbiosis between legumes and the root nodule bacteria called rhizobia that supply them with their nitrogen. And this research led him into the area of evolutionary biology. So, what's evolutionary biology? I suppose we're going to hear more about that tonight from Ford. But basically, I think his book, it presents an entirely new approach to the challenges of food security and sustainability that draws on the principles of evolution. We all, we think we know a little bit about evolution and, how we have evolved and how plants have evolved and so on. But what lessons can we draw from that for our current modern plant breeding and indeed animal breeding programs? In his book, Ford shows how biotech and traditional plant breeding, they can use Darwinian insights to identify promising routes for crop genetic improvement and, equally importantly, avoid costly dead ends. The book, and I'm sure he'll touch on this tonight, it reveals why it's sometimes better to slow or even to reverse evolutionary trends when they're inconsistent with our present goals, and how we can glean new ideas from natural selection and the marvellous innovations that has delivered in our wild species. So I'm sure they're the kind of aspects that Ford is going to cover tonight in his lecture. And I'm not going to try to predict what he's going to cover, but before I stand down and let him at it, I just want to give you a few quotes that I saw from some reviews of his book, because it has, as I said, exercised a lot of discussion and debate and comment in the scientific community and beyond. So a quote from Science, the highly respected science journal, is that Darwinian agriculture offers an engaging and bold explanation of why agricultural research must take better advantage of insights from evolutionary biology. Another quote from the journal Biology: Darwinian agriculture shows just how much plant breeding and biotech can learn from evolutionary biology and takes an honest look at agricultural technologies from both ends of the spectrum, from genetic engineering to organic farming. So he steers a course that touches on both of those extremes of technology. So there are many, many other reviews of the book that are available. I think the worst one I saw forward was that said, this is a very interesting book that will stimulate a lot of discussion. So if that's the worst review you get, I think you're starting from a fairly positive place with regard to the lecture that you're going to deliver to us here this evening, Ford. So I'm very much looking forward to your presentation here, and we will have a chance for a debate and a discussion involving yourselves after that, and I think we will have a very stimulating and interesting evening ahead. Thank you.
Thanks, Frank. And of course, Ireland and Minnesota have very strong links. The city of Minneapolis is actually older than the city of Belfast. I don't know how many people know that. But the links, of course, Minnesota is a Scandinavian and Irish state. I once asked a professor, one of your colleagues in the U of M, why there were so many Irish in Minnesota, since it's so cold, and the climate is definitely harsher than it is here. And he explained, he said, the reason is very simple. He said, when the Irish emigrated to America en masse, they were pushed further and further west by other immigrants, and they ended up beyond Chicago in the Twin Cities and in the prairie, the wonderful, wonderful prairie land of Minnesota. And they would write back to their friends in Ireland, and they would say, this is a wonderful land of milk and honey, and would make absolutely no mention of the cold winters. And the letter, in those days, it was a bit like email is today, or Twitter, in fact, very like Twitter. The letter would be passed around the community at home in Ireland, and everybody would read this letter about this wonderful land of milk and honey where the sun shone all the time. And so more and more immigrants from Ireland went to Minnesota. But we're very pleased to have an eminent academic from Minnesota, originally, of course, UC Davis, with us tonight. I call now on Professor R. Ford Denison to deliver the first of these lectures.
Well, thank you. It's certainly an honor to address such a distinguished group. The title of my talk today, Evolutionary Trade-Offs as Opportunities, is also the title of I think it was a 2010 paper that I wrote in Evolutionary Applications, which I believe is available free online, so you have an opportunity to check some of my references and so on by referring to that. Or even better, you could buy a copy of my book. I would encourage you to do that. The graph that I show because of the opportunity to make a brief mention of Thomas Malthus. Now, both Charles Darwin and Alfred Russel Wallace, the co-inventors, if you will, of the theory of evolution by natural selection, they both credited Malthus as providing a key idea that exponential growth of populations leading to shortage of resources was a major factor driving natural selection. And the exponential growth of populations is a pretty easy thing to predict mathematically, and it is indeed what we tend to see. But the other thing that Malthus said was that food supply in food production by humans could be increased, could be improved, but only linearly. And of course, an exponential growth will always overtake and surpass linear growth. Now, this linear growth always seemed a bit odd to me. I mean, why should, what's the law of nature or principle of physics that would cause food supply to grow linearly? And yet, that does seem to have been the recent pattern, as shown on the slide. Linear growth, but linear growth sometimes running into an apparent yield plateau, as you see in the top three lines, which are wheat production for three European countries. These are data for like 1960 to 2010 or something like that. And so, while I think the general public is not aware of it, those of us in agriculture tend to be pretty concerned about whether it will be possible for food production to keep up with increasing demand, both from population growth and economic growth and diversion of agricultural products into ethanol and other products. So that's basically what I'm focusing on today. I'm going to cover maybe 30 or 40% of the material in the book, because I have 40 minutes to speak, and if you're interested, you can have a look at the book, or you can get the table of contents on the Princeton University Press website and see what topics I haven't covered today. But I'll be talking mainly about opportunities for genetic improvement of crops and the implications of past evolution. Now, when I say that I've written a book called Darwinian Agriculture, most people assume, incorrectly, that my main focus is about the problem of evolution of insecticide resistance in pests, or as shown here, herbicide resistance in weeds. But I'm not really showing this slide for that reason. What I've got here is watergrass, or Echinocloa. It's a serious weed of rice in California, where I worked for 12 years. And the highlighted rice plants come from a couple of organic farms, and you can see that those highlighted watergrass plants come from a couple of organic farms, and those plants, because they hadn't been exposed to herbicides, or their ancestors hadn't been exposed to herbicides, were readily killed by these two different herbicides. You can see they're dead. Whereas here in the back row, we have watergrass from conventional farms that had been exposed to these two different herbicides and actually evolved resistance in only a few years. Only a few years, because herbicide resistance is a relatively simple trait, as anybody at Monsanto can tell you. It's a relatively easy thing to bring about. But I'm going to give a couple of other examples of the evolution of watergrass because we know its history. We know that it evolved from barnyard grass in only the last few thousand years in Asian rice fields, because prior to that we didn't have much in the way of flooded rice cultivation in Asia. So what did it do during that time? Well, barnyard grass has this red stem, and so farmers could spot an invading weed with this red stem from 100 meters away and were immediately out there and removed it. And it wasn't until Echinocloa evolved a green-stemmed mutant shown in the middle that it had much chance of persisting. Now that would have been enough to fool me because I don't work that much with rice, but rice farmers, of course, noticed other differences between Echinocloa and rice and were quite active in removing these plants, and so these weeds, and so they inadvertently, over a few thousand years, imposed additional selection on Echinocloa to look more like rice so that they didn't see it in an escaped cultivation. Until by the mid-1970s, when Spencer Barrett assessed a bunch of visible traits of barnyard grass, watergrass, and rice, he found, as shown in this statistical plot, that Echinocloa in rice fields had actually evolved to look more like rice than it looks like its own ancestor from only a few thousand years ago. So relatively rapid evolution. That wasn't the only challenge, of course, for this invading weed. Rice fields are typically flooded, and barnyard grass is not very flooding tolerant, and so it was pretty well controlled and sort of, growing on the edges where the water was shallow. Here at this is the Rice Research Center at UC Davis, and the dark green are the deep water plots where there were very few weeds, and then you can see in the shallower plots more weeds. But as I mentioned, the ancestor of watergrass was not flooding tolerant, but in this same few thousand years, it evolved both these erenchyma, these air-filled channels that let oxygen diffuse down into the roots in the flooded soil, and a gas-impermeable layer, highlighted here, that keeps the oxygen from leaking out before it gets to the roots. Now, I'm going to use an example later in my talk of drought resistance. I've been told that that's not actually much of a problem here in Ireland, and I probably should have put more emphasis on flooding tolerance. But in any case, I'm using this to illustrate the rapidity with which resistance to abiotic stress can evolve under natural selection. So this leads to the central argument of my talk. I argue that if a few thousand years was long enough for Echinocloa on a relatively limited land area, a few thousand years was enough to evolve flooding tolerance and quite accurate visual mimicry of the crop. And if you then consider that natural selection operated on the wild ancestors of our crops for many millions of years, they were exposed to drought, they were exposed to pests, they were exposed to low nutrient soils, they were exposed to high winds, cold, all the various forms of stress. You have to imagine that all of the relatively simple meaning arising frequently through mutations, all the relatively simple solutions to these many problems that plants faced must have arisen repeatedly over the evolution of the wild ancestors of our crops. And those solutions that worked the best, those were the plants that, the genotypes that took over the populations, and those traits were then inherited by the wild ancestors of our crops, and to some extent carried down today through land races and modern cultivars. So my argument then is that for the kinds of traits that the wild ancestors of crops were exposed to, the kinds of stresses that wild ancestors were exposed to, it's going to be quite difficult to make further improvements in those kinds of traits. Now, this doesn't apply to things like herbicide resistance because they don't have a long history of exposure to herbicide resistance, so it's not hard to introduce improvement in that trait. But for things like abiotic stress, I argue that there are going to be two approaches that are going to work. What's not going to work is, taking some gene that they already have and cranking up the expression of that gene. That's the kind of thing that arises all the time by random mutation. Natural selection has tested that option over and over and over again, and there's a reason that it didn't stick. Similarly, changing an amino acid or two in the active site of an enzyme, that's the sort of thing that happens all the time by mutation. So it's unlikely we're going to come up with something that hasn't already been tried that way. But what we can do, the focus of this talk, is often evolutionary trade-offs, trade-offs that limited some trait that benefited a plant under one set of conditions but decreased its success in another set of conditions, that kind of a trade-off. Those trade-offs that were rejected by natural selection aren't always agronomic trade-offs. They're not always trade-offs from our point of view. And in the cases where what we want is different than what natural selection was selecting for in the past, that's the low-hanging fruit, the easy things for us to improve. Now, the other option, which I think will be more difficult, but in the long run also quite valuable, is biotechnology coming up with really brand new radical innovations, things that are so different from anything that's out there in our crops today that it's never arisen over their evolutionary history, never been tested by natural selection. And I'll give a couple examples of that as well. But my focus is on the trade-offs. So obviously trade-offs can be constraints. If a plant has a given amount of nitrogen, which is a key ingredient in protein, you can breed for high percent protein, but that's also breeding for low yield. Or you can breed for higher yield, but that's also breeding for lower protein, all else being equal. But there are many cases where trade-offs may be opportunities, and the four cases which I will discuss with examples of each. First, trade-offs in adaptation to past versus present conditions. Second, trade-offs between the individual fitness or individual competitiveness of individual plants and the performance of the whole field of a crop, where they may all be the same genotype, not competing against different genotypes. Trade-offs between the fitness of symbionts and their host plants, that's my own specialty. And finally, it may be, lost here, trade-offs between early reproduction and longevity. That would probably be the most speculative part of my talk, but maybe stimulate some thought. And the bottom of this slide is a visual index of these four cases. You'll see those again. So I'll start with trade-offs between past and present conditions. This graph is not labeled past versus present, but I'll get to that. This is the trade-off for different rubiscos. This is the photosynthetic enzyme rubisco, and the vertical axis is the activity. Greater activity means you need less of the enzyme to get a given amount of photosynthesis. And the horizontal axis is the specificity of this enzyme to interact with CO2, its desirable substrate, rather than oxygen. When it interacts with oxygen, we get photorespiration, and we basically have some lost yield from that. So starting here with all the data that I have for crop plants, we see what looks like a very strong trade-off, negative relationship between these two desirable traits, specificity and activity. If we broaden our perspective, which we can do in the age of biotechnology, where we can perhaps transfer traits from unrelated species, we can broaden it to include bacteria and red algae. And back in 1999, the journal Science had one of those articles about how, we have these yield plateaus and things are looking bad, but don't worry, biotechnology will solve the problem. And they're going to do it by transferring the Rubisco genes from red algae into our crops, because Rubisco has this greater, in red algae, has greater specificity for CO2, and that will reduce losses to photorespiration. Well, I did point out in our 2003 paper on Darwinian agriculture that at least for the red algae, that they did have lower activity, and so there was a trade-off there that you would need more Rubisco, you would need to put on more fertilizer to have more leaf protein to get the same rate of photosynthesis. But since then, people, including some biotechnology folks, have looked at this issue again and pointed out that because atmospheric CO2 has increased and is increasing, they wondered whether our Rubisco, where it might lie on this curve, and whether it's at the optimum position on this trade-off curve given present levels of CO2. Because we know that often, usually in fact, evolution lags behind. If things are changing, evolution will change in response, but often it lags behind the present conditions. And so Zhu et al. Actually calculated what would be the optimum place on this trade-off curve for crops. And they found that present-day crops are at the optimum place for atmospheric CO2 of 200 parts per million. But today, atmospheric CO2 is 400 parts per million. So the optima actually has moved to the left, and so they're suggesting that we could actually perhaps use a bacterial Rubisco or engineer the Rubisco in our crops to give up a bit of CO2 specificity, because CO2 is increased concentration. Specificity is not as important. It's still good, but it's not as important as it was. And so they're basically advocating, and I think they make a reasonable case, for doing exactly the opposite of what biotechnologists were proposing in 1999. That's not the only possible contribution from biotechnology, though, because there are, in fact, cyanobacteria that have broken this trade-off, apparently. Now, they do have quite low specificity, the opposite of what was considered desirable in 1999, but they have dramatically higher activity. So perhaps with this Rubisco, we could have much lower leaf Rubisco. Well, are there any agricultural situations, though, where leaf protein is important? This may actually be something important in Ireland, where you have, cattle that are eating leaves, and actually a large fraction of what they're eating in those leaves of that protein is Rubisco. So it's not necessarily something that would be good for forage crops, but perhaps for grain crops. Let me move on to my second category of trade-off opportunities. This is actually an area where there's already been a significant amount of work, but I think there's also still untapped potential. And this is the trade-off between the individual plant and the plant community interests. This was laid out brilliantly in the 1968 paper by Colin Donald, an Australian agronomist. Many of you have probably read this paper. Read it again. It's worth reading again, believe me. The paper is best known for coining the term idiotype. Unfortunately, that's all that some people remember, but the main point of the paper was the hypothesis that there's a negative relationship, that is a trade-off, between individual competitiveness of plants and their collective yield when you have a whole field of the same genotype. And Colin Donald went on to propose a long list of specific traits which would sacrifice a bit of individual competitiveness, but if you had a whole field of that genotype, you would have higher yield or higher resource efficiency. And these traits included short stature, relatively erect leaf angle, and reduced number of reduced branching that has a single stem in the extreme. Now, a couple years before Donald's paper was published, the originator of IR8 rice, I'm blanking on, Peter Jennings, also the same name as a broadcaster, but Peter Jennings, the plant breeder, laid out his design for a high-yielding rice variety, and he predicted, without as much sort of a theoretical background, that short stature and erect leaf angle would be key to high yield. And he then went on to introduce these traits into rice and got IR8, the first Green Revolution rice, shown here, flanked by its two parents. You can see how much shorter it is, so it's transgressive segregation, I guess, but it also had relatively erect leaves and dramatically higher yield potential than materials that were available, including its own parents. Well, similar things were seen in wheat. This is, each of these points here is a wheat cultivar. These are released over a period of decades, and they were analyzed by Austin et al. The vertical axis is yield, horizontal axis is height. So you see this strong negative relationship with the high-yielding plants being the shorter stature plants, exactly as Colin Donald predicted. All very well, but the second part of Donald's hypothesis is that these high-yielding materials, now, he made it clear that a low competitive plant wouldn't automatically be high-yielding, right? It might be highly disease susceptible. That would be uncompetitive but also low-yielding. But that every high-yielding plant would be low competitive. So that was actually tested by Peter Jennings for rice. That's the dashed line. They mixed high and low yielding rice together, let them compete against each other, took the resulting seed, replanted, repeated that for 3 years, and in 3 years the high yielding materials had all been outcompeted by the low yielding materials. This actually repeats something that had been done by Sunison decades previously with barley with the same result. Now I'd like to turn, many of Colin Donald's ideas have already been implemented by plant breeders, but I'd like to suggest that there are some still opportunities remaining for exploiting this trade-off between individual competitiveness and whole crop yield or whole crop resource use efficiency. And this is one that I worked on many years ago when I worked for U.S. Department of Agriculture, the phenomenon of solar tracking where leaves turn to follow the sun. So this leaflet of lucerne or alfalfa that I've highlighted has a slightly higher photosynthesis rate because it's facing directly at the sun. But this highlighted leaflet has a lower photosynthesis rate because the leaf that's shading it is facing the sun and therefore casting a larger shadow. So the question is, what's the net result of solar tracking on photosynthesis of a whole crop canopy? And to answer that question, we built this outdoor sunlit photosynthesis chamber, measured photosynthesis with the leaves in their normal tracking orientation, and then there's actually an internal turntable that allowed us to turn the plants and disrupt solar tracking and see whether photosynthesis went up or down. The journal that I submitted the paper, The Effects of Crop Rotation on Photosynthesis, objected to the title. But here are the results. So the vertical axis here is the ratio of photosynthesis by the whole canopy with the plants in their natural tracking orientation divided by the photosynthesis when their tracking is disrupted by turning. And so anything above the dashed line is a positive contribution from tracking. Anything below is a cost of tracking. And so you can see that solar tracking increased photosynthesis under low leaf area conditions when the shadows cast by leaves were predominantly falling on soil rather than falling on other leaves. Although it was a small, I mean, we never saw more than this 5% benefit, not much. And then as you went up to higher leaf area and leaves are increasingly casting their shadows on other leaves, we see this negative effect at high leaf area index. But this is, okay, so I've highlighted the benefits and the cost. But there's a little bit more to this story, because if you look at our experimental model, you see that it's a bit questionable, because the lower leaves here in this 1 square meter plant community are exposed to light from the side that they wouldn't get if they were in the middle of a field. So we've got this edge effect, and this extra light that the lower leaves get would reduce the cost of shading by upper leaves, biasing the results in favor of solar tracking. And so I also explored this question with a computer model. Of course, you know, you can't really believe computer models, but here it is. And it suggested that if you include this, if you don't have this artifact of edge effects, solar tracking actually has a photosynthetic cost over a wider range of leaf areas. And that over the season, modeling it using data and harvest frequency for Davis, where I had the data, there's actually a 5% yield cost to solar tracking. Well, why would natural selection, over millions of years, I've said that in just a few thousand years it can do these wonderful things. It had millions of years to eliminate solar tracking, and this has to be an easy trait to get rid of, right? I mean, any number of knockouts, mutations, null mutations are going to knock out solar tracking. Now, if I gave you the task of taking a non-solar tracking plant and making it track the sun, that would be a challenge. You'd need a bunch of genes. But to get rid of solar tracking, any number of genes, you knock them out and it's gone. So why did natural selection not do this? This shadow is a plant shading its neighbor. It's not shading one of its own leaves. And that's generally the case. And anytime natural selection has an opportunity to reduce the photosynthesis of competitors coming up in a plant's shade by 50%, and it only costs the shading plant 2 or 3% of its photosynthesis, it's going to take that deal. Because if that competitor gets up there, it's going to be competing not just for light, but for water and nutrients and so on. So the species as a whole, well, if you had a field of nothing but solar tracking alfalfa, or a field of non-tracking alfalfa, and good weed control, that would be key, the non-tracking, I think, would have significantly higher yield. But it's easy to understand that natural selection based on individual competitiveness would have missed this opportunity to improve community performance. Here's another example of that. Corn and maize, many other species, produce much more pollen than is needed to pollinate all the female flowers. Darwin referred to this, the incredible waste of pollen by our fir trees. And the thing is, there's a cost to making these overlarge male flowers, a direct carbon and nitrogen cost, but also I've highlighted here that these tassels shade lower leaves. And my first postdoctoral advisor pointed out, Bob Loomis, pointed out that this is actually a significant photosynthetic cost, this shading by corn tassels. If you fly over Iowa during anthesis, you're looking down and seeing yellow. That means the sun is falling on non-photosynthetic pollen grains, not on leaves. Now, why would natural selection have missed this opportunity to improve this trait? Well, I really blew it here. I swear at this point, I usually show a picture of the Irish elk. But when I was asked to, limit my talk to 40 minutes, I took that out, not thinking, where I was going. But it's exactly the same thing, that the overlong antlers of the Irish elk were bad for the species as a whole, but if you didn't have long antlers, you didn't get a mate. Species went extinct, oh well, natural selection has no foresight. Now, abiotic stress, I mentioned the example of flooding tolerance, which should be something I understand logically that's of great concern with winter crops here in Ireland. But the big emphasis of the biotech companies, in the US at least, has been on the opposite thing, drought tolerance. And so the question is there a lot of opportunity for improving water use efficiency, that is the amount of yield you can get from a given amount of water? And this graph from Angus et al., Ted Schau, one of my former colleagues at Davis, was the senior author on this paper. The vertical axis is yield corrected for the biochemical composition of the seed, because some seeds take more photosynthate to make a high protein seed than a high starch seed, for example. And then the horizontal axis is the evapotranspiration, the water use by the crop. And so the ratio of those two is the water use efficiency by one definition. So basically what this trade-off curve shows is that you can get more yield by using more water, but you're basically moving along this curve. There's doesn't seem to be any significant intrinsic difference among these crops in their underlying physiological water use efficiency. That changes dramatically if you include plants with the C4 photosynthetic pathway. And this is why people at the International Rice Research Institute and elsewhere, with funding from the Microsoft Foundation, or Gates Foundation, I guess it's called, have been putting a lot of energy into trying to develop a C4 photosynthesis rice. I think this will be a very challenging thing to do. Now, it's not so challenging that evolution couldn't do it. Evolution's done it, dozens of times. We know that the C4 photosynthesis is in all these different pathways of the plant evolutionary tree. So we know it's evolved independently many times in the plant kingdom. But nonetheless, it's much more difficult than anything that biotechnology has been able to achieve to date. But I think if they do it, they probably will get rice that is substantially higher in water use efficiency, quite possibly higher yield. And it's not obvious to me that there'll be major trade-offs. So I wish them luck with this. But meanwhile, this is what we've got from the two leading biotech companies. The green line is the yield of a Czech variety, the blue of the latest release from Pioneer on the right that was developed to be drought tolerant. And the two The two sets of bars at the right are the yield trial of this pioneer material under non-drought conditions at the right, where they had sort of similar yield, and then moderate drought in the middle, where they also had somewhat similar yield. So it doesn't look like it was a big advance. The Monsanto data show a small apparent benefit under severe drought to their tolerant material. But one thing that's worth noting is that the Pioneer data are from an independent test of Pioneer's material by university scientists. The Monsanto data come from a paper in PNAS with only Monsanto authors and with very, very little information on the details of how these field tests were done. So, they don't give any of the kind of thing that Agronomy Journal would insist on, for example. Meanwhile, I think the low-hanging fruit for increasing water use efficiency may come through this trade-off here. This isn't strictly speaking a trade-off curve. This is water use efficiency, here defined as the ratio of photosynthesis to transpiration over the daylit period of a day. And so, as you'd expect, the plants get more photosynthesis, or rather use less water for a given amount of photosynthesis when the temperature is cool and humid in the morning than in the afternoon when it's hot and dry and the water is rushing out of their stomata when they try to photosynthesize. So something that I think would be worth trying. Now, this may already have been done inadvertently by plant breeders selecting for water use efficiency. I'm thinking specifically of the very impressive work of Robert Roberts in Australia. But if you had a crop which had some kind of stomatal closure or partial stomatal closure in the afternoon, then it would use water only when it could use water most efficiently, that is in the morning. And a given supply of water in the soil would last longer into the drought or longer into the terminal drought in the Mediterranean climate. And so closing the stomata in the afternoon would reduce photosynthesis per day. But by making the water last longer, you would have more days of photosynthesis and potentially greater seasonal photosynthesis and yield. And that's what matters. We don't care how much a plant photosynthesizes today. We want maximal seasonal photosynthesis and maximal yield with the available supply of water. Maybe not too relevant to Ireland, but I think similar arguments might be applied to the kinds of abiotic stress that you see here. Now I'll turn fairly briefly to my own specialty of symbiotic nitrogen fixation. And if you look at a bunch of different strains of rhizobia, the root nodule bacteria that provide nitrogen to clover here in Ireland, the vertical axis is yield. In this case, this is soybean data. The horizontal axis is the cost to the plant, a proxy for the cost, the nodule mass it has to make to get that yield. And so the star is USDA 110. That's a widely used inoculum strain. You can see it has the highest yield benefit and only a moderate cost. But all these other strains, which tend to be much more abundant in nodules, unfortunately, than USDA 110, are not useless, most of them, but provide perhaps only half the benefit that USDA 110 does. So if we could somehow have all of the nodules on the crop occupied by USDA 110, we would cut in half the cost to the legume of fixing nitrogen. We could either double the nitrogen fixation or cut the cost in half and free up photosynthate that could be used by the plant for other purposes. As something of an evolutionary biologist, I wondered, well, why? Why is there all this variability? You know, natural selection, millions, this is a very old symbiosis. It's had millions of years to come up with the best genotype and eliminate all the losers. Why is there this variability? And the answer, I hope you can anticipate by now, is trade-offs. And let me explain. The electron micrograph at the left shows a cross-section of a root nodule infected by USDA 110, very active in fixing nitrogen. I've circled 3 of these cells that are cranking away, turning nitrogen gas into ammonia, but they're also taking a little bit of the malate or succinate that they get from their plant host and stashing it away as this storage lipid, polyhydroxybutyrate. You can see these little tiny white crystals. I think you can see them in those black dark cells. Now, when Hahn and Studer did a simple knockout of the nitrogen-fixing enzyme, that's the genotype at the right, or the nodule at the right, all of the energy that was going to nitrogen fixation, a lot of that ends up going into this PHB, and you can see these huge PHB granules, white granules, in this non-nitrogen fixing, non-beneficial, isogenic strain. Well, my grad student Will Ratcliffe, now on the faculty of Georgia Tech, showed that a high PHB rhizobium cell can not only survive for a year with no external carbon supply, but they can actually double or triple their numbers, divide once or twice, just using this internal resource that they've hoarded when they were in the root nodules. So this suddenly flips the situation on its head. Rather than asking, well, why hasn't past natural selection made these rhizobia great nitrogen fixers? Looking at this, we have to ask, the rhizobia don't care whether the plant reproduces or not. They're not transmitted in the seed, they're transmitted from dying nodules. And every individual plant has 10 different genotypes on average of rhizobia. So you have this massive tragedy of the commons where no one strain has any real incentive to keep the plant from dying. If they can divert resources to PHB, they're only shortchanging the plant of 10% of its nitrogen because there's 10 strains, but they've got all this extra resource. So now we have kind of the opposite question. Why haven't rhizobia all evolved to be virulent parasites of legumes? That's actually what the model of Stuart West, who did a sabbatical with me, he's now a famous evolutionary biologist at Oxford. He was starting to get famous already. He's not famous because of me. But his model said that nitrogen fixation, this trait should have disappeared under evolution very quickly. But it didn't. Why not? And so, because it's been around for millions of years. So the hypothesis we came up with is that the plants don't let them get away with it. And what we hypothesized is that somehow legumes must monitor the nitrogen contribution from each individual nodule, all of them containing different strains, and nodules that don't deliver enough nitrogen get shut down in a way that prevents reproduction, further reproduction by the rhizobia inside. So this experiment, shown at the left, we grew the plants up in normal air with nitrogen gas, so they provided nitrogen during the early growth of the plant. But then we put these little gas chambers around the individual nodules, and some of them we gave normal nitrogen, so they fixed nitrogen at 100% of their potential. That's the right side of the graph. Some of them we gave no nitrogen or just enough to fix their own nitrogen needs. That's the left side of the graph. And then Toby Kears did this experiment with soybean. She's just made full professor at the Free University of Amsterdam. And Ryoko Ono did it with lucerne and pea and got similar results. And she's just started at UC Santa Barbara. So they measured the reproduction of the rhizobia in the nodule, and that's the vertical axis here. So if you just contrast the very active nitrogen fixers at the right with the quite poor nitrogen fixers at the left, at the right you have the nice pink legume nodules that we all know and love. If you don't know and love them, dig up some clover and look at the nodules and you'll see how pink they are. Unless you have bad rhizobia, then they're going to be white or brown or blue. And at the left, we have senescing nodules because the plant is shutting down these nodules that are fixing at 0, 17, or 33% of potential. Now, to get 33% of potential, you have to decrease the nitrogen concentration to 2 or 3%, I forget. But as still having a bit of a, 1 foot in agronomy, what I'm really concerned about is these rhizobia in the middle, the ones that are occupying not lots of nodules out there and only contributing half as much nitrogen relative to their cost as the best inoculum strains. So that's these guys here that we artificially reduced their nitrogen fixation to 50% of potential. So they had a lot of resources to stash away as PHB or to otherwise use for their own reproduction, and yet their numbers were not statistically significant below those of the most active nitrogen fixers. So it looks like most of the time, rhizobia can divert half of the energy they get from their host into their own reproduction without triggering the host sanctions that reduce their reproduction or survival. Now, it's easy to understand why rhizobia, if they could get away with diverting half of their energy into their own reproduction, why that would be favored by natural selection among rhizobia. But legumes have been evolving for millions of years too. Why didn't evolution in the legumes give them stricter sanctions so that they would only tolerate the very best local strains? They would kill all the mediocre, not just the non-fixing rhizobia, which, not surprisingly from this, are rare, but they would kill all the mediocre rhizobia in their nodules and lavishly reward the very best indigenous strains, subsequently releasing them into soil in vast numbers so that their offspring, so that future generations of legumes growing in that soil would have only great rhizobia. Why did natural selection miss that opportunity? And this is the same trade-off between the interests of the individual and the interests of the community. Do you ever see that in society, that kind of trade-off? I've seen it in agriculture and have some examples in my book, actually. From the standpoint of the species as a whole, they could all be hollering, kill those mediocre rhizobia. We don't want our offspring to have to, have those bad rhizobia in their nodules. But from the standpoint of the individual plant that needs nitrogen to complete its, seed protein needs, the benefits of supporting mediocre rhizobia from an individual plant's point of view may well exceed the costs. But just as in the case of plant breeders reversing natural selection in breeding short but high-yielding wheat and rice, reversing natural selection in taking out the bitter genes in cucumber, reversing natural selection and getting rid of thorns in blackberries. You know, all these cases where we've reversed past natural selection. Plant breeders could make a different choice than natural selection did in the evolution of the legumes. And this is kind of a cartoon of how this might be done. You start out as usual in the top row with a genetically diverse population of soybean, or here, clover, and measure the seed yield or forage yield, but don't base your selection just on that first year yield. If this is a crop that's going to be grown in rotation, you would then grow in the same soil a genetically uniform test crop of that rotation species. So in this case, in the Midwest, we rotate corn and soybean, maize and soybean, and so this second year, this soil that's been affected by the first year different genotypes of soybean gets a single genotype of corn, but two of these maize plants grow bigger because, hypothetically, the legume genotypes 2 and 4 preferentially enrich the soil with a mycorrhizal fungal species that was particularly beneficial to corn. Soybeans do this, actually. This has been demonstrated by Nancy Collins Johnson. But the question is, do they differ, do genotypes differ in the extent to which they enrich the soil with maize benefiting mycorrhizae? She actually hypothesized that a good piece of the rotation effect may be due to this effect. Interesting hypothesis. And then, either in year 2 or in the case of rotation in year 3, we're now going in with a genetically uniform test crop of the legume. They're the same genotype, but one of them is growing bigger because, hypothetically, genotype 2 in the first row had really strict sanctions and enriched the soil only with the very best rhizobia from the mix that were in that original soil. Now, maybe that's not what it was. Maybe genotype 2 somehow enriched the soil with disease-suppressing bacteria, or somehow depleted the population of pathogens in the soil, or enriched the soil with some nitrogen cycle bacterium or nitrogen cycle inhibiting bacterium that had a beneficial effect 2 years later. That'd be okay, right? I mean, our main idea here is that we haven't really thought, at least most plant breeders haven't really thought, about how the genotype we plant this year affects the growth of next year's crop and the crop after that. We think about that for species. How does, this year's species affect next year's crop? But I think it hasn't been done that much for genotypes. So I'm saying breeding for benefits to future crops in the same soil is something that natural selection doesn't do because it has no foresight, and I think it's an untapped area for improvement. Relatively untapped, at least. Has anybody done it? Ask me during the question period. Yes, thank you. So I will go fairly quickly through my last and most provocative, speculative point, and that's the trade-off between reproduction and longevity. And this actually came about from an agricultural context. I directed the first 10 years of the long-term experiment at UC Davis, shown here in the upper right. And so when I came to Minnesota, they asked me to be an advisor to our relatively new 3-site network of long-term experiments. But the most cited paper from the long-term work at Davis was this work showing that organic tomatoes, same variety of tomato, grown side by the same people under the same weather, have double the level of this secondary plant compound, camphorol, which is thought to have beneficial effects on human health. Well, why organic? The thing is, most of these chemicals are actually defensive chemicals. They're things, toxins that the plants make to protect themselves from insects or pathogens or nematodes. So then the second part of the question is, well, why should some natural insecticide be beneficial to human health? First, I thought this was just a weird coincidence. But then all these other examples started coming in. You know, natural toxins in broccoli or kale that seem to be beneficial to health, things in garlic, and so on. And so Matson and Cheng actually pointed this out, that there are all these natural toxins, defensive toxins in plants that are apparently beneficial to human health. And their hypothesis was that these toxins at low doses, the toxicity effect is negligible, but they trigger natural defenses that are beneficial. Well, fine, but if these defenses are beneficial, then natural selection would long ago, if they're always beneficial, if there's no trade-offs, natural selection would long ago have made them constitutive rather than inducible by these plant toxins. So what's the trade-off? Best known trade-off in evolutionary biology is the trade-off between longevity and early reproduction. Here, this is actually shown for one of these secondary plant compounds, resveratrol, which greatly extends the reproductive lifetime of nematodes. I mean, these nematodes are still reproducing at the ripe old age of 7 days, whereas the wild type, or the untreated rather, have very little reproduction then. But here's the trade-off. To get that, they have to give up a little bit of reproduction early in the first 3 days. So here's the question. Not yet. Is this true of humans? Not clear. Results are a bit contradictory, but these data for British aristocratic women are certainly consistent with this. Those that lived into their 80s or 90s, those were the ones that had their first baby in their mid to late 20s rather than their early 20s. So maybe there's this trade-off between age of reproduction and longevity in humans. If this is true, would we be willing, if it gave us a few more years of healthy life, would we be willing to sacrifice just a little bit of teenage pregnancy? The thing is, natural selection wouldn't. And that's shown by this model here, where I look at 2 genotypes over a number of generations, modeled, and there's basically, a couple of famines here. You can see those dips there from the famines. And I assumed a trade-off between early reproduction and longevity, but even assuming that trade-off, the dashed line genotype, which always reproduced later and therefore had slightly lower risk of dying, they disappear from the population. They're losers in the evolutionary race relative to the early reproducers that get the compound interest effect of having, children, grandchildren, and great-grandchildren compressed into fewer years. But these are not the only 2 options. You also have the possibility of an inducible response, delaying reproduction under some conditions but not others. And that's what I modeled. That should be just about right. That's what I modeled here, a genotype that when conditions are good, they reproduce early, they track the reproduction of the early reproducing genotype, but when conditions are bad, they still die off, but because they delay reproduction, well, half of them delay reproduction based on a cue which I'm about to tell you about. Based on that cue, half of them delay reproduction. They get a bit of a trade-off. They don't die off quite as fast. And if you go through a couple of crash boom cycles, they've doubled their representation in the gene pool. And what's the cue? Oh, I guess I put it on the screen. The cue is famine foods. If you're eating leaves, it's probably because, if your ancestors were eating leaves, it's probably because they couldn't get meat. And that meant that a famine was coming, and that meant if there's a trade-off between reproduction and longevity, that the fitness optimizing strategy was to delay reproduction if delaying reproduction increases your chance of surviving. It's actually, if there's a big population decrease, it can actually be better from an evolutionary point of view to produce 2 offspring next year rather than 4 offspring this year. If those 2 offspring are joining a gene pool which is only 10% as big, then they're a larger fraction of that gene pool. And the individuals that delay reproduction in response to a cue that population is going to decrease, they end up being the ancestors of a larger fraction of future generations. So obviously, this is something that needs to be tested both in experimental systems and in humans. But it does suggest the potential that if we give our bodies cues of population decrease, that may decrease our intrinsic fertility only a small amount, not necessarily enough to keep a family from having the number of children that they want. They might just have to have sex more often, a sacrifice that many people would be willing to make to achieve the desired family size, but perhaps getting a few more years of longevity. So this is just a summary slide, and maybe I should stand down. Thank you for your attention.